So yeah, hello everyone. I'm Jeffrey Jacobs from the Hasselt University, a PhD researcher there, and I will present today you some findings and results from our study, if green roofs can compensate for the loss of biodiversity in cities. And today I will focus on the Hymenoptera group. So first, an introduction about urbanization. Recent projections indicate that the percentage of the human population living in urban areas worldwide will reach nearly 70% by 2050. This will have a major impact on the biodiversity of our planet, as an increase in city area causes loss of habitat for species, an increase in spatial distance between remaining pockets of green, and a change in habitat quality. The climate in urban areas is also characterized by the so-called heat island effect, where cities tend to be hotter than the nearby rural areas. As you can see in the figure below, where we see that in the city center it's up to 4 degrees Celsius hotter than in a nearby rural area. On top of that, the increased amount of pollutants in cities will influence species directly or indirectly via food quality and quantity, and urbanization has a lot of negative effects on human health. All these negative effects from urbanization are of most concern for species that are threatened and economically important organisms such as pollinators. So by 2030, more than 25% of all endangered species will be affected directly or indirectly by urban expansions. And we see that worldwide coastal areas are places with a high degree of marine and terrestrial diversity because of the high habitat diversity. Cities are often concentrated alongside these coastline and major river systems. As you can see on the figure, you see that the orange parts are areas with a high diversity, and the blue dots are cities, expanding cities worldwide. And we see that a lot of the cities worldwide are expanding in those areas with a high diversity. So to counter these negative environmental effects of urbanization on nature, the construction of green roofs is often promoted to increase the amount of green space in cities. Green roofs could deliver several important ecosystem services, otherwise provided by natural green. They not only add an aesthetic value to the city, but studies show benefits for water runoff and evapotranspiration, reduction in the heat flux due to a better insulation, and a beneficial role in carbon sequestration. Previous studies have shown that green roofs are an efficient solution to mitigate the previously discussed heat island effect. And in addition, green roofs can increase habitat connectivity by acting as stepping stones. Moreover, especially during the last 2 decades, studies have indicated that green roofs located in large cities have a high potential to support species that are negatively impacted by habitat loss. The most common type of green roof, at least here in Western Europe, are so-called extensive green roofs, consisting of a 5 to 20 centimeter deep layer of a homogeneous shallow rocky substrate. They are typically planted with allochthonous plants, such as species of sedum or other drought-tolerant plants, as they require minimal maintenance and are adapted to summer drought. Arthropods and other microorganisms begin to colonize these green roofs almost immediately after installation, because they are often brought in by the planting material. In the figure below, you can see a cross-section of an extensive green roof with all the different layers present. According to the island biogeographical theory by MacArthur and Wilson, species richness in isolated habitat fragments is determined by isolation and surface area. Green roofs can be seen as green islands in the sky, surrounded by inhospitable surface area, where both the size of the roof and the distance to other green areas could have an impact on species richness. So therefore, we hypothesized that green roof situations closer to the ground level will have a higher diversity and abundance of Hymenopterans. And we also hypothesized that green roofs with the larger surface areas will have higher species richness, because larger roofs can be more diverse in microhabitats. Species use spreads to maximize their fitness. In a changing environment, we also expected a change in species composition due to dissimilar roof features, such as the vegetation composition. So for our study, we sampled macroinvertebrates on 20 green roofs in the city of Antwerp, Belgium. The selection of the roof was based on several parameters found in literature, like the surface of the roof had to be at least more than 25 square meters. The roof had to be at least 3 years old, and so on. We sampled from March till September 2020, and we used pen traps to assess the flying insects communities on the green roofs. 4 different colors of pen traps were used, as you can see in the picture down below. They were set on each roof and left for a period of 24 hours. We emptied the traps and invertebrates were stored in 70% ethanol. Ground-dwelling invertebrate communities were sampled with so-called pitfall traps. The traps were rain protected, as you can see on the figure down below, with a plastic lid. And on each roof, there were 4 traps installed and left for approximately 3 weeks. Invertebrates were collected after those 3 weeks and stored in 70% alcohol as well. All the specimens that we found were sorted into higher level taxa and are still being identified to species level. In both traps, we use propylene glycol, as it is a sticky agent that does help to catch the invertebrates and does not damage the DNA of the collected specimen, as some other sticky agents used in the field do. So for our green roofs, they have an average height of 10.5 meters with a range from 4 to 23 meters, an average surface area of around 300 square meters, and an average depth of the substrate of 7 centimeters. We calculated Simpson and Shannon-Wiener diversity indices for each roof. And most of you will know, but I will shortly describe these indices. The Shannon-Wiener index value increases with biodiversity. In contrast, the Simpson index measures the relative abundance of species and is therefore more of a dominance index. To check if, like we expected, there would be a change in species composition due to the vegetation cover, we divided the green roofs into 2 groups. One group with roofs mainly consisting of species of sedum, as you can see on the top picture, and one group with roofs mainly consisting of species of sedum, herbs, and grasses, as shown in the picture below. So during our sampling period, we captured around 23,000 specimens. As you can see in the table, they are listed there with their respective groups. And we try to identify as much individuals to species level as possible, as said before. So far for the Hymenoptera, we identified all the ant species, around 3,800 specimens, and we found 17 different species. And all the wild bees, 326 specimens with 22 different species. And we have a preliminary result of 850 wasps identified to species level. If you look at the diversity indices per roof, the first graph when looking at the Formicidae, the ants, and the second graph when looking at the wild bees, we see that the diversity indices do vary between the roofs. While the wasps are not yet included, as we do not have all the results present. Some roofs have a diversity index close to 0 in the graph, but it only means that there is a low number of species present. This does not say anything about their abundances. For example, in the graph on Formicidae, we see that ECO1 only has a very low diversity index, but in fact we found 2 species there, with one species, Lasius niger, being super dominant with more than 1,000 specimens. For the wild bees, as for the ants, there were some roofs where we did not find a single specimen of ant or wild bee. We have not yet been able to explain why on these roofs we did not find a single specimen, and are still trying to figure this out, which roof characteristics do influence these numbers. When we compare the 2 groups, so group 1, roofs planted with sedum, herbs, and grasses, and group 2, the sedum roofs, we see that there is a difference in Simpson and Shannon-Wiener index, but differences were present where we did not find them to be statistically significant. We're not going into depth on all the results as time is limited, but we have found no statistically significant difference with the height of the roof, not for the diversity index and not for the species richness. And we did not find a significant difference with the surface area of the roof. So our results do not support the island biogeography theory for species of Hymenoptera on our green roofs that we sampled. So some conclusions. As mentioned before, we found differences in diversity indices between roofs, but we could not explain them statistically with the height, surface area, or vegetation cover of the green roofs. Nonetheless, we see small differences. Other factors beyond the scope of our study, for example, sunlight exposures, as some roofs have more sunlight exposure than others, could have an impact on the diversity indices, and further research is needed here. When compared with results from ground level habitats, as we also sampled green spaces in the near proximity of these green roofs, which is less than 100 meters from the green roofs, we see that species richness for Hymenoptera that are quite similar between ground level habitat and green roofs. However, we see slightly lower abundances on green roofs compared to ground level, presumably because of differences in provided habitat. Nonetheless, we believe that our research shows that green roofs can be a suitable habitat for Hymenoptera groups in cities, but further and more detailed research is needed to see if they can really compensate for the loss of biodiversity in cities. Thank you for your attention, and I'm looking forward to your questions.
Thank you so much, Jeffrey. That was great. We are just trying to stay on time here today. There is a question in the Q&A box. Jeffrey, if you'd like to pop in there and maybe answer it, type an answer. Save us a little bit of time. That's great. Problem. Thank you so much.
Thank you.